Before we get into the bulk of the material in this chapter, it might be useful to review some things that we've already taken a look at. One thing is you might want to go back and review the Conning-Gold prelog rules or designating the RNS configuration at a chiral center. This is something that was covered in Organic Chemistry 1. Then the structure and reactivity of carboxylic acids, so lots of chemistry associated with carboxylic acids. Also, nucleophilic acyl substitution, which is also in the carboxylic acids chapter. And of course, properties of amines, right? We're going to be talking about amino acids, which have amino groups in them, and they have carboxyl groups, right, to make a carboxylic acid. And so we're going to be discussing both of those. Now, throughout the textbook, there's these biolinks boxes. And in those biolinks sections, if you read them, you would have explored the relationship between the structure of a compound and its medicinal activity. And the relationship between structure and medicinal activity is perhaps the most striking for biological molecules that we call proteins. And I'm sure many of you have heard about proteins in biology classes. And if you've ever wondered, what is a protein exactly on the molecular level, it's nothing more than a polymer that's assembled from amino acid monomers that have been linked together, just like the jigsaw puzzle pieces that you kind of see here. You see these amino acid pieces here, right? And then they click them together, so to speak, and I'm just being kind of silly here, but you see how they're kind of putting these puzzle pieces together? Well, if you link enough of these amino acids together, you can make a protein. So again, all a protein is, it's a bunch of amino acids that are bound together. Now, if you're astute, which I know you all are, you probably noticed that the amino acid has an amino group, right? And it also has a carboxylic acid, hence the name amino acid. The amino acids that we're going to be interested in our class are called alpha amino acids. And an alpha amino acid means you have an amino and a carboxyl group separated by only one carbon, okay? So if we have a carboxylic acid, okay, if you just permit me to kind of scribble one down here, if we put a carboxylic acid, right, the first carbon off of the carboxyl group, that's called the alpha carbon. And so if we put an amino group attached to that, that's where we get alpha amino acid. And you can see that at the alpha position, we can also have an R group. And so you see that drawn right here. Now, if we go back to this R group, you can see here that as long as the R group is not a hydrogen, right, because we know there's a hydrogen here that's implied, right, there's a hydrogen here that's implied. I could have put it in here as well. So if this R group is a hydrogen, then it's not chiral, and then in that case, it would be glycine. So when your R group is hydrogen, it's glycine. So if you want to memorize that, it's not a bad idea to do that. But if the R group is anything other than hydrogen, then your amino acid is going to be chiral. And so we're going to be talking again about alpha amino acids in this chapter. And if the R group is a hydrogen, it's going to be achiral, and that means the name of that alpha amino acid is glycine. And if the R group is something else, then it's going to have another name. For example, when it's a methyl group, it's called alanine, et cetera, et cetera. And so we're going to take a look at the structures of the alpha amino acids. Now, when you start linking the amino acids together, okay, before you get to a protein, if you have only two amino acids linked together, we would call that a peptide. So it says here, a relatively short amino acid chain, so less than 40 or 50. For example, if you have two amino acids linked together, again, that would be a dipeptide. If you had three linked together, it would be a tripeptide, tetrapeptide, so on and so forth. If you get up to, in the 40 or 50 range, then you have a polypeptide, and then above that, you're going to have a protein. Now, this word peptide that you keep hearing me say, a peptide bond is nothing more than an amide bond. I repeat, okay, when you hear me talk about a peptide bond, it's just an amide bond. The only difference is a peptide bond is an amide bond that connects amino acids. So you see here we have a tripeptide. Well, it's not really a tripeptide because, they're showing part of a bigger molecule. But I digress. You can see here that the amide bonds here, that's what we call a peptide bond. So again, a peptide bond is nothing more than an amide bond between amino acids. And then again, and if we have a larger chain of amino acids linked together, that's what we call a protein, okay? And protein implies that there's some kind of biological function associated with the molecule.